Alright, this is going to be our last video for chapter 12, and it's going to be over events at the synapse. And so when we're talking about the synapse, so you have two neurons, or you have a neuron and a muscle cell, but it's essentially going to be the junction between two cells, and this is going to be the location where they're communicating. So the synapse is just that junction, it's that space between these two cells. So as I mentioned, it could be a neuron to neuron, like you see here. You have the axon terminals of one, and then you have the dendrites of another. So the presynaptic cell is the first cell that's bringing the information in. It's transmitting the signal. This will be the one that's ending in the axon terminals here. And then you have the postsynaptic cell, which is the one that comes after. So like in the picture here, we have the dendrites of another neuron. So this would be your presynaptic cell and your postsynaptic cell. Now this could also be an effector cell like a muscle or a gland, okay? So there's actually two types of synapses. You have electrical synapses. Now this is not the one that we're going to be focusing on because electrical synapses are much less common and they are actually a direct connection. So in the case of an electrical synapse, the cells are actually directly connected by special junctions. A lot of times they're gap junctions. And so they can pass these ions directly from one cell to the other. And if this cell is depolarized, it's going to trigger depolarization in the other cell. It's a lot faster, but it's a little more difficult to control. And there's going to be certain areas in our body where we want to find these electrical synapses. One example is in our heart. You will find some. But most of our neuron junctions are going to be chemical synapses. And this is going to involve turning that electrical signal into a chemical signal, which is going to be neurotransmitters. So here you can see that these two cells are not directly connected, they're not touching. And so we have this synapse, or this gap in between, and the action potential is not able to jump or move directly across that gap. And so we're going to have to turn that electrical signal into a chemical signal. And so when we're talking about the events at the synapse, we do want to make sure we understand that we're focusing on a chemical synapse. So once again, that action potential is going to reach the axon terminals, and that's what this is, just a blown up picture. So this would just be one of your axon terminals here. This is what we call our presynaptic cell. And when that action potential reaches the end, it's actually going to trigger voltage-gated calcium channels to open. And so calcium is greater on the outside of the membrane, and so what's going to happen is calcium is going to enter the cell. So when that action potential reaches those axon terminals, it's going to cause calcium to enter the cell. Now what the calcium is going to do is it's going to kind of interact with these little vesicles. And these little vesicles are just membrane-bound sacs. And inside of them we can see these little dots. These represent our neurotransmitters. And so the calcium enters into the cell, and it's going to interact with the membranes of these vesicles. And it's actually going to trigger these vesicles to move to the membrane here, this axonema, and it's going to cause it to release those neurotransmitters by a process called exocytosis. So the vesicles are going to move to the edge of the membrane, they're going to fuse with the membrane, and they're going to release these neurotransmitters into what's called the synaptic cleft, which is this gap. Now once the neurotransmitters are released into the synaptic cleft, they're actually going to diffuse across to that postsynaptic cell. And they're just moving by basic diffusion from high to low. And over here on the postsynaptic cell, we have some ligand-gated ion channels. And so remember that ligand-gated ion channels, they only open when a ligand or a chemical messenger binds. So in this case, we have our neurotransmitters. That's going to be our ligand, or our chemical messenger. And so when the ligand binds to these channels, it triggers those sodium-gated ion channels to open. The neurotransmitter binds to the protein, and then that channel is going to open up. Now, as we know, when the channel's open, sodium is higher on the outside, so sodium is going to rush into that postsynaptic cell. And when sodium rushes in, that's going to cause depolarization of that membrane. So it's going to cause a change in the membrane potential, and essentially it's going to lead to depolarization here, which will then be carried forward. Now, we're not going to just continue to stimulate the postsynaptic cell, because kind of like with the action potential, it's quickly moved along and then it goes back to normal. The same thing is going to happen in the synaptic cleft. These neurotransmitters have short-term effects because, number one, they rapidly diffuse, and so they just continue to spread out, so they're no longer concentrated in the area. They could also be broken down by enzymes, and the enzymes are always specific. So like in the case of acetylcholine, which is what's going to bind to our muscle cells, there's an enzyme called acetylcholinesterase, and what it does is it breaks down acetylcholine into its component parts, choline and acetic acid. This choline actually gets taken back up and turned back into acetylcholine, back in those axon terminals. But these neurotransmitters are not staying in the synapse. They're diffusing and they're being broken down, and so those effects are going to be diminished once that concentration goes away. And so here we have the presynaptic axon terminal. So once again, action potential comes along here. It causes calcium to rush into the cell. The calcium is going to cause those vesicles to come in and release their neurotransmitters. And those neurotransmitters are going to diffuse across the synapse and bind to proteins on the postsynaptic cell. And sodium is going to rush in, and if it's enough of a stimulus, it's going to trigger another action potential like what we've seen already. So over here we just see these chemical messengers. Here's some examples of neurotransmitters. We talked about acetylcholine, that's responsible for like muscle contraction. But we also have others like adrenaline or epinephrine. We've got dopamine, serotonin. So each of your neurotransmitters is going to be specific to a certain pathway. And so that's going to wrap up chapter 12. So we do have some videos here. There's also some videos that are posted on Moodle, but this particularly are crash course videos. One is over action potentials and one is over the synapse. And so I would encourage you to watch both of those. And as always, we have completed the chapter, so it's important that you make sure you've done all of your activities and your quizzes on Moodle. And please pay attention to due dates. Be sure you have your test taken by the final date. And hope you guys have a great day.